Okay, all right. So let's get started. Hi, everybody. I'm Kimberly Ruckman. I work for NeuroNeeds. I do their social media management. And today I'm here with Dr. Richard Bowles, who's going to be giving us a presentation on genetic testing in autism, results in precise diagnosis in 2/3 cases, implications for personalized therapy. Take it away, Dr. Bowles.
Okay. First of all, these are the objectives that I want to get over to explain, to learn about how to use whole genome sequencing. WGS is the way it's abbreviated often. How it can provide really precise diagnostic information in most people with autism, about 2 out of 3 cases. To understand that most of those cases were not on the laboratory report and require additional review of the DNA. Learn that the whole genome sequencing just doesn't give you a diagnosis, but can also help with treatment options. In fact, there were treatment options in about 2/3 of the cases. And understand that the genetic testing really greatly improves our understanding of what autism really is. Explains mechanisms behind many of the therapies that are used by biomed people, including the MAPS group, and that it can help patients that don't even have genetics testing just because we have a better idea what autism is. So I'm going to go over a case in which the genetics made a big difference. There was a 13-year-old girl. Previously, she had developmental regression in the 1st grade and recovered. She was given a diagnosis of autism. She had also other issues such as auditory and visual processing delays, but she was almost at grade level in all classes and was doing actually quite well before at age 12 she got just a common cold. And in one day, she lost all of her academic abilities. Her behavior regressed, became almost catatonic. She acted like she was an infant, and then had unusual seizure-like events. The EEG was always normal, but these events were preceded by headache with muscle weakness, was more on the left than on the right side, often with balance problems. That's ataxia, rapid heart rate. She was pale, incontinent, clammy, and often with nausea. She also had some functional issues. She had migraine, chronic pain, chronic fatigue, and POTS, or dizziness on standing. And she had a diagnosis of common variable immunodeficiency with frequent infections treated with IVIG and high-dose steroids. So the diagnosis clinically is hemiplegic migraine, episodic ataxia which is caused by a calcium channelopathy or a defect in a calcium channel. The prolonged episodes of headache with confusion, ataxia, balance problems, and other neurological problems, including asymmetrical weakness, one side being more than the other, is really consistent with and highly suggestive of hemiplegic migraine. A hemiplegic migraine is due to mutations in calcium channel genes, and sometimes in the pumps that use ATP to pump sodium and potassium across the membrane. These are the 3 genes that are most often mutations in these genes cause hemiplegic migraine, and these are the mutations in this girl. She had a mutation in all 3 of them, which explains why she had an unusually severe case. Interventions were just the mitochondrial cocktail. That's no alcohol in it. It's a cocktail. Things are put together. Multiple vitamins and minerals, antioxidants, other natural compounds, cofactors, etc., to help the mitochondria make energy and to detoxify it. Potassium chloride, and one drug, acetazolamide, which is really a diuretic that's used to remove it. It helps potassium go into cells as well as other things. And on this treatment alone, she had a really dramatic improvement. In February 2020, her academics, she had improved somewhat before this, but she was tested at kindergarten in reading and 1st grade in math. A year later, same test, 8th grade in reading and 7th grade in math. She was in the 8th grade at the time. So you can see that she went from being actually substantially developmentally delayed intellectual disability to having normal ability on the right diagnosis and testing. Also, her behavior, which had been severely regressed, became age appropriate. Her balance problem went away, and many of the other problems improved. Still has some chronic pain and fatigue, but is really like 98% better than where she was before. So what this case is really showing is that if you have an exact diagnosis, you can often make a tremendous difference with therapy. These are some of the hats that I wear. I'm coming to you today as the Chief Medical and Scientific Officer of NeuroNeeds. It's a company that produces products for neurological disorders, including the mitochondrial cocktail that helped with that patient. But I'm also the director of the neurogenomics program at Neurabilities, and it's under that I see patients, including patients with autism, and do genetic testing. And that's predominantly what I'm going to be talking about today is genetic testing in autism. I also have been a medical director of DNA sequencing companies and learned how the ins and outs of DNA sequencing. And then I'm a researcher. I had a laboratory at USC, Children's Hospital Los Angeles. I'm still writing papers. In fact, the data that I'm going to show you is just submitted for publication. So this is the machine that can do it. If you remember, the Human Genome Project is the first time that the entire DNA code of a person was read, and it took more than 10 years. Laboratories all over the world cost multiple billions of dollars. This is the machine that can read a human genome in 3 hours. This part here is the size of a refrigerator. It gives you an idea of the size of the machine, and it does cost $1 million, but it's quite amazing. It can read 3 billion nucleotides pairs 30 times over, because to make sure that there's no errors in a few hours. Things have progressed so incredibly rapidly that you really can't look over many years to say, well, what can we do now? Because every year is a lot different. The amount of DNA sequenced, exome sequencing, which a lot of people get, it sequences the genes. It's only 1% of the DNA. Whole genome sequencing, which I'm going to be talking about, is over 99% of the DNA. There's still tiny areas that are hard to do. So it's not just the amount of, and a few years ago we did exome, and a few years before that they were panel testing and then individual testing. So we do more than 1,000 times more DNA testing than was done even 5 years ago.
So Dr. Bowles, question. If we had parents who have their children's DNA done through Cortigen, should they redo it?
Oh yeah, that's more than 1,000 times more DNA is now done. Okay. And the other advantage is it's not like, well, what will be next year? Well, we're doing over 99% of it. We don't have that much further to go. Even though our ability to understand the DNA or understanding of the biology increases year by year, it's on the computer, and if it's sequenced now, you can then later on with better understanding figure something out that you might not be able to do now. But there's also the accuracy, the computing power, using the computing power for interpretation, number of cases reported. All of this has made a tremendous difference in just in years. So that what I'm going to be talking about now are the last 50 cases done, and there has been really no major innovations in this in the last year and a half in which these 50 cases were done. So this is the paper that has been submitted for publication. So it is available now as a preprint. If you want to actually see the paper, it's highly technical, but your doctor might be able to get something out of it. You just go to preprints.org, and then under the author name, put my name, Bowles, B-O-L-E-S. And you can see all my preprints, and then this one will be one of them. And so this is the paper that it's not even peer-reviewed yet, so it's not yet published, that I'm going to be showing you the data. So this is the 50 most recent patients with autism that I personally evaluated. They were all done at Neurabilities, which is a healthcare organization in the Philadelphia area in New Jersey and Pennsylvania. But it was with telemedicine, so they were all over the country with a few cases abroad. And 75% of them are Caucasian, but the other 25% are highly diverse. They all had trio. That's the patient, however, you say the child, but the child sometimes an adult, plus both biological parents, which is necessary to find new mutations, de novo mutations that I'll get to. And then this is the timing in which the actual sequencing was done. And it was following the last major update to the interpretation software. And then a few other things. Qualifying variants, what do I mean by that? Well, people have tens of thousands of variants. What I mean by a qualifying variant is, first of all, that it's verified. I went into the back end of the computer and we verified this variant is real. It's not a mistake of the sequencing or of the computer. That it's a coding, it changes the amino acid code, it changes the protein code. The vast majority of variants do not change the amino acid code. Less than 1% of them do. The variant had to be very rare. For almost all of them, it had to be less than 1 in 10,000 chromosomes that you see it. For recessive, because you have to have 2 copies, it was less than 1%. There was a few of those. And conserved, that means it's rare in humans, it's rare in other organisms. If you look back all the way through vertebrates down to lamprey, which are more primitive than fish, if every single species has exactly the same amino acid in that location, it's probably a very important part of the protein, and changing it probably makes a difference. On the other hand, if every different species has a different amino acid there, it doesn't matter what your child has in that position. So I wanted to make sure that every single one of them was evolutionary conserved. So that meant qualifying variants. To get a primary diagnostic variant, it had to be qualifying, but it also had to have data directly related to autism. And those are some of the ways in which that was done. So it wasn't just a variant in any gene, but a variant in a gene in which it had been published with a direct autism association. And all of this means that if it's called a PDV, a primary diagnostic variant, that variant is very highly likely not only to be related to the autism in that patient, but to contribute the bulk of the genetic causation, meaning that it's probably more than 50% of why that patient has autism is due to that one sequence variant. So here is the clinical data. I know this is very complex, and it's really, I'm just having you look at the color. First of all, the participant number, the first 25 is here, and then the last 25 is here. So all 50 are on this slide. Here is the numbers here is the age of the patient. If it's pink, it's a female. The whites are male. It's a 4-to-1 ratio, which is what you see in autism. Here is the developmental status, how much intellectual disability. High-functioning autism generally means that their intellectual disability is mild, or they have normal intelligence, or even advanced. Low-functioning autism has significant intellectual disability. You can draw the line anywhere you want, but I drew the line at moderate, because moderate intellectual disability and less generally requires assistance throughout the lifespan, living at home, an institution, something like that, where other ones generally are in the population, working, having family, etc. And so the ones that are in yellow are the more severe, and the ones in white are the less severe. It's exactly 50-50. So while this may be a little bit skewed towards higher ones than maybe what you would see in a school or something, this is 50% of them are mild and 50% of them are severe, which is pretty representative of what you would see in any neurological practice. Verbal. Some people with autism have almost no verbal ability. Sometimes they can have normal intelligence. Oftentimes they don't. If they're essentially nonverbal, they're in green. Otherwise, they're in white. So you can see that there's quite a few that are nonverbal, but most are not. The olive here are seizures, spikes on EEG, but no clinical seizures, was mild here. But you can see that seizures are common, but a lot of them don't have it. The regression, neurological regression, that you've lost skills. If it was only once, it's mild, and then the more the tan that's darker is they've had a very severe or multiple episodes. You can see some had no episodes of regression, some had mild regression, and some of them had more severe regression. And then everything else that's neuropsychiatric is in this one here. The light blue, it happens to be tics, which is often you see tics in more of a pandas-like thing. There's really not a line between autism and pandas. All of the patients in this study had to have met diagnostic criteria for autism, but that doesn't mean that pandas is not what's going on. Pans are pandas. So this is, you can see that these patients are representative of patients in general with autism. Most of them, or all of them actually, have additional problems, and that they're really not particularly skewed in any direction, but what you would see in pretty much any autism practice. Here is the genetic data. So this slide really has pretty much all of the data on. I know that it's really complicated. There's a few things I want you to see, and it's mostly the color. First of all, the first 25 here, the second 25 here, 26 through 50. The ones that are in blue here have a first-degree relative with a significant neurodevelopmental disorder. And I define significant as it affects their life in some major way. And so you can see most of them don't have a major family history, but quite a few of them do. This is the one here. This is what we found on the inheritance pattern. The yellows are predominant. You see, the yellows are exactly 25 out of 50 have yellow, half of them. That's a de novo mutation. That is a variant that's not in either parent, that is in the patient. That variant is not only in the patient, but it is a primary diagnostic variant. It's real, it's rare, it's conserved, and it's in a gene that's directly related to autism. In addition to that, to get a primary diagnostic variant, they had to fit the inheritance pattern. For a de novo, because it's not in the parents, basically the family history is negative. But occasionally we'll find, like this one has a sister with ADHD. Well, ADHD is really common and probably due to other issues going on. So half of the cases, the cause of autism is a new mutation not in the parents. That's 50%. Another 20% is a variant which was inherited from one or both parents in a Mendelian manner. X-linked, there are 3 of them. They're the pink ones here. And each of them, the mother was an unaffected carrier. Autosomal recessive, that's where both parents are normal carriers and the child got a double dose, double copy. There's 4 of them because there's 2 over here. 2 of them where the child got the same copy from both parents, and then the other ones the child got a different mutation from each parent. And then autosomal dominant, that's directly from a parent, an affected parent, to an affected child. One came from dad, one came from mom. So and then there's one here that's matrilineal. This one here was on the mitochondrial DNA from mom. So 20% of them were inherited. 50% of them had an inherited variant which is causing autism. 50% of them was a de novo variant, so that's 70%, but one patient had both, a mitochondrial DNA and a de novo. So it was 68%, which is just over 2/3. We were able to make it a precise diagnosis based on the DNA. So de novo variants. Again, those are variants which new mutations that aren't in the patient, that are not in the parents. There were 42 de novo variants in all of the patients everywhere, regardless of what the variant is, regardless what the gene was, that actually changed the coding sequence. Of the 46 de novo variants, 31 were in genes directly related to autism, and almost all of them, 29 out of 31, were qualifying. If you look at so those are 31 in genes related to autism. If you look at 15 in genes not directly to autism, only 33% of them, this is actually should be 5 out of 15, were qualifying variants. Almost all of them were actually in genes indirectly related to autism. That means they're related to a gene that's related to a gene related to autism. It's a little bit confusing here, but basically the odds ratio is about 30. That means that if you have a de novo variant, you're 30 times more likely that that variant is related to autism than it's just there and it's not related. And it's very highly statistically significant. This would come up by chance alone only 2 in 10,000 times. So what this is saying is that, and it's very complicated, but that the vast majority of the de novo variants, well, 28 out of 29 would be directly related to autism, and one of them not out of 29. Also, we looked at the de novo rate. If you look at what's published throughout the DNA, it's about 0.2. That's 0.2 de novo variants per person. Or in 10 people, you would see 2 of them in the average person. So they do occur in people that don't have disease, but they're reasonably rare. Most people don't have them. If you exclude the ones that we think are related to autism, our patients had 0.24, which is very, very close to that. So they had a basic background rate that's not associated with autism, same as other people. But on top of that, they had all of these variants here that are probably causal for autism. Okay, so I went before that 68% had a primary diagnostic variant, and that means that that variant is highly likely to be related to autism. Most of those were de novo. Some of those were inherited with different inheritance rates, different inheritance models. Okay, here's some more stuff. This is exactly the same data as you saw before, it's just arranged differently. These are the ones that the mutation that I found that I believe is the cause of autism, the laboratory put it on their report. When they send the report to the doctor, that was somewhere on the report. And these here, the ones in green, it was on the report. The ones in orange, what I found to be the cause of autism, was nowhere on the report. So even though it was on the report had a different thing. So this was the color of the report. 7 of the reports, there are exactly 28 here. And I'm sorry, there are 14 here. And of these 14, 7 of them were red reports. 5 of them said positive. This is the cause of disease, and 2 of them were likely positive. It means that they highly think that this is the cause of the disease, but they can't say 100%. So if you include the likely positives, there were 7 of them. That's 7 out of 50, so that's 14%. So on the laboratory report, only 14% of them said it was positive or likely positive. The others were uncertain or candidate. That means, well, we found something, you should take a look at it. And then these are green. They were on the laboratory report, but the lab, for whatever reason, thought that they were probably not the cause of disease. So if you just look at the laboratory report and you just look at the positives, only 14% give a diagnosis. If you then add in these variants of uncertain significance, there are 10 of them, so 20%. So 14% if you only look at the reds, and 20% if you look at the reds and the yellows. And then there's another 8% or so that show up here. So the vast majority, and remember, all of this together is only 28%. So all of these were known disorders. The laboratory is only looking for known disorders. The laboratory is not looking for new diseases. It's not in doing research. That's something I do. So every single one the laboratory put on its report, even if it thought it was negative, was a known disorder. Now these are the ones the laboratory didn't put on the report. You can see there's 4 of them that were a known disorder. But they're very rare known disorders. And the laboratory in general did extremely well. So it did, out of 18, it found 14, which is actually pretty good. Remember, I had people working under me, and we spent hours on each case and went into it pretty much into depth. And we didn't find that many more known disorders than the lab found. There were 3 that were very rare. Very rare means 1 to 4 cases in the entire world. Those are not known disorders. That's, oh, we found a case with a mutation in this gene with autism, and it may be an autism gene. The lab isn't doing that. That's not what they do. So there were 3 of them that had been seen before, that had been published before in autism, but they were very rare cases. All of the ones here in orange are novel. That means this is the first case in the world. If you look at the human genome database here, most of these have at least one neurodevelopmental disorder that have been seen before, but they're not published. That's just saying some lab somewhere in the world found a variant in this gene in a patient with neurodevelopmental disease. That could be a completely benign variant in somebody with ADHD. You don't know anything about it, and there's nothing published about it. So most of these have been seen before, but not published. So out of the 50 patients, we found 14 novel disorders. In other words, 14 new diseases that haven't been seen before. So 28% of the cases had a disease that had never been seen before. If you add these in here, fully a third of the case have never been seen before, or only a few in the literature. Another thing I want you to look at is that you see, well, these don't overlap at all. Every single one of these is different from every other one. That there was no patient that the same gene was found in twice. This is what I say, there's thousands of autism genes. It's really, each and every one is different. It's not like you look for this gene or that gene. If I did another 40 patients, you'd probably find another 40 genes. So there's really a very large number of genes which can cause autism. The rest of this part here is really the mechanism. Okay, so there's thousands of genes that can cause autism. That's the genotype. And autism can show in 1,000 different ways. Every patient is different. But there's a bottleneck in between. There's sort of like an hourglass. In between is the pathways. There's not that many pathways. This is what the protein does. I know this is very complex, but it really fits into these 7 pathways. Cation transport. Cations are positively charged salts like potassium, sodium, and potassium, sodium, and calcium. Redox state, that's the electrical charge of it that has to do with mitochondrial energy metabolism. Amino acids. Ubiquinization, that's a protein degradation pathway, how old proteins are destroyed. Neurotransmitters that send signals from one nerve cell to another. Gene expression, that's turning on and off the DNA to read other genes. And then cell division, how one cell divides into another. But anyway, these 5 right here are treatable pathways. So of the 7 pathways, these 5 are treatable. This one, it turns on and off other genes and it might be treatable, but it's really hard to know exactly what it turns on and off. Some of these are treatable. And then the cell division one is really not treatable. But 5 of the pathways are treatable, and some of them, like in particular redox state, cations and neurotransmitters and amino acids, are actually quite treatable. So while autism genetics is very complex, most of the patients have treatable pathways. Not all, but most. Okay, so this is what I got on before, that 14 of the 34, only 41% were on the laboratory report. By reanalyzing the raw data, you can go from 28% to 68%, but it's only 14% if you only include the positives and the likely positives. 28% if you include it's on the report anywhere, including likely negative. But after reanalysis, I went to 68%. And then the ones that were quote unquote missed by the laboratory, most of those are new diagnoses not seen before. Labs are not, that's not what they do. And then there were a few that were extremely rare and wouldn't expect them to have found that. Okay, this is the same one you saw before, but now we're going to look at this over here. This is a pathway. This is one here that we didn't look at before. So these are de novo here, the de novo genes that are in the yellow. These are inherited genes related to autism. The ones that are in italics, like the FOC2 here, nice name, are related to the diagnostic. That's the X-linked one here. But almost all of these are not in italics, and they are not primary diagnostic variants. These are all variants that are qualifying. They're real, They're rare and they're highly conserved. And they're all in genes directly associated with autism. But they didn't meet the criteria for a primary diagnostic variant. In other words, what these are is that these are likely related to autism as risk factors. They don't cause it, but they contribute to it. And you can see the vast majority of the cases have them, and many of them have many of them. The average is 2 per patient. So polygenic inheritance, more than one gene. Yes, 2/3 of the case have a major genetic factor. That one mutation or variant is the lion's share. But cases with and without a primary diagnostic variant have these candidate polygenic modifiers. And autism is complicated. It is not just a single gene in most people, but these other modifiers. We know there has to be modifiers because there is not a single gene that a mutation causes autism in everyone. There's not a single mutation in any gene that everyone has it. Even if you look at things like Fragile X or other ones that are very, very highly likely to cause autism, Angelman syndrome, etc. Some people have autism and some people don't, even though they all have brain disease. So we know that there's got to be other factors involved, and many of those other factors are genetic. There's also environmental components as well. We know there has to be environmental components because at least half the children with autism, they develop their problem after a deterioration. That's what I was talking about in the first one, about they had some sort of event, some sort of regression. And usually there's an environmental component. Infection is most common, but there are many other things that I've seen before. I've seen cases that had surgery or medication or anesthesia or even an immunization. There are many other things that can be the thing, but there's almost always something happened and then the child deteriorated within hours afterwards. And so there has to be an environmental components as well. Okay, so these again are back to the pathways. The 7 pathways, 4 of which are potentially treatable, and 1 of which, well, I treat the ubiquitization disorders. Most people would think they're not very well treatable, but I treat them with mitochondrial cocktail, and they seem to respond. So this is why when I get to the second part here, well, if it's all genetic, what can you do about it? Well, the genetics tells you what to do about it. Okay, actionability. All right, you found what it is. What can you do about that? The DNA changed what I did in 37 out of the 50 cases. So 13 of the 50 cases, okay, this is what I found. I'm not doing anything differently. Sometimes we were on the right path beforehand. I used mitochondrial cocktail in everybody. If it was a mitochondrial thing, that would have been one of those 13. Okay, we're already on the cocktail, nothing more to do. So that doesn't mean they weren't treatable. That just means that the DNA did not change what I did. But 3/4 of the time, the DNA did change what I did. In particular, 2/3 of the time, 33 of 50, I made treatment changes. And you can see that 30 of them were non-prescription supplements, that's dietary supplements over the counter, and 24 were prescription medication. Obviously, this does not add up. Most of the patients got the most common patient got both a prescription and a non-prescription recommendations. So many times there were, and most of them didn't have just one, but I think the average was maybe about 2 to 4, somewhere in there in terms of recommendations based on the DNA.
So Dr. Bowles, I have a question. So if this is genetic based in a child, and you say, you know, that they require a supplement, is that a supplement that they would have to take their whole life? Or is that something changes as they get older?
It would presumably be, if it works, it would be something that you'd want to continue for life, because otherwise the genetics is there, and if you take away the supplement, they'll go back to where they were. So yes. Okay. So there's no easy cure on this. We can treat, but we can't cure autism at this point. In most cases, we can treat it. A lot of times people are afraid that you know, you do the DNA and they're going to find 10,000 things wrong. And a lot of insurance companies don't want to pay for it because they think it's going to open up Pandora's box and we're going to do all these new tests and that's going to cause a lot of problems. only 6 of the 50, the DNA said I should order another test that was related to the autism. So only 12% did I do any other testing. 88%, I said, well, this is it. We don't need to do any testing, or we didn't find anything. There's nothing else to do. And then people are afraid that, you're going to find things in every pathway and you're going to send them to every specialist available. Only 9 of the 50, or 18%, did I refer them to a specialist. Almost all of those were to immunology. Now, immunology was because of 2 reasons. Either A, I found a gene in an immune system. I found a variant in an immune system gene, and I want the immunologist to determine how to treat it. Or B, I didn't find anything, and so I assume that this is an immunological cause of disease. Most of those looked like PANs or PANDAS. And if the genetics is clean, then it probably is immunological, or at least it might be immunological. So I sent them there to figure out if they can find it. So remember, there was about 1 in 5 cases that I didn't find a primary diagnostic variant, and I didn't find a whole bunch of secondary variants that I thought were the cause of autism. So maybe about 20% or so, I really didn't find that much, and then they were sent to immunology. Okay, off target. Off target is not to do with their autism. You find something that's completely unrelated. 4 of them, that would be 8%, found a pathogenic. That means it causes disease in the FLG gene. FLG is a very common cause of skin disorders. It can cause all sorts of problems. Patient I just saw yesterday, which isn't in this study, I found a mutation in the FLG gene, and she gets a bunch of skin problems where she has her orthotics rub on her skin. And when you put tape on her for an IV, you pull the tape off, her skin comes off, and no one can figure out why. But she had a pathogenic variant in FLG, meaning the skin falls apart easily, because FLG is what holds the cells together. It's the glue between the cells, the mortar between the bricks. It's partially that. And if you only have 1 copy that's working, you're going to have less glue and the cells fall apart more. If you have 1 mutation, you get eczema and dry skin and flaking. And if you have 2 mutations, you can get a much more severe disease, but that's very rare, like ichthyosis. 4 of them had an increased cancer risk. This is what people worry about. But 2 of them had a mild increased risk. They were in the APC gene. It was a common mutation in APC that might increase the risk of cancer a little bit. So they were instructed to talk to their primary care provider. Maybe they should have a colonoscopy or mammograms maybe 5 years earlier or something like that. It would be up to the doctor, but really not much of an increased risk. 1 of them knew about that before, and 1 of them did not know about that risk. There were 1 other with a previously known mutation. There was a mutation in MUT-YX, and the family knew about that was running in the family, and that was a cancer gene. 2 of them had a mutation in von Willebrand that suggests that there might be increased bleeding. 1% of the population has von Willebrand. You want to know about that before surgery. Blood testing was done in both of them. 1 of the cases had von Willebrand, and that's important if the patient has a car wreck or surgery. The other patient, the testing came out negative. There was previously identified deafness gene in 1. That means that there's a mutation in a gene that the patient was known to be deaf, and we now know what the mutation is. And 1 of them had mosaic Turner syndrome. They already knew that, but the testing found that, which is already known. And then another patient found, it was a teenager with a very significant sleep disorder, had a mutation in PER3, and that predicts a sleep disorder, and that might be helpful for the sleep doctor to know what's going on. So basically, bad news. You found something that they didn't know about before that really makes a difference. There were only 2. There was another one here. See, the 4th one with cancer, increased cancer risk due to BAR-D1. That's a significantly increased cancer risk. But now they can get screening, like colonoscopies, mammographies early. Insurance should pay for that, so hopefully we'll find cancer early and may even save the patient's life and other family members as well. And then 1 had a variant that suggested vascular Ehlers-Danlos. A lot of testing was done, all of which was negative, by the way, but this did lead to additional testing. but only 4% had really what you would consider to be bad news, that things the family didn't know about. So it can happen when you do DNA, but it's really very rare. 68% get a diagnosis, 4% get bad news. This is all the stuff that I went in before. This is all just kind of putting it all together in one slide about what this all means. So if we have the slides available on the website, I'll go over this. Okay, the end of the presentation, that's the end of the DNA genetic sequencing presentation. Now I'm going to try to make sense of it as to how you should treat, a child differently with autism. These are reef sharks in the Bahamas, and I am right next to the person with the camera. I didn't take this photo, but I was just to the right of the photographer. They're actually very playful sharks that you can teach them to do tricks for food. Okay, cations, positively charged salts. That's one of the main pathways in autism. That's not really that recognized in the literature or by physicians. What happens usually is that the channels are leaky, and so the cation, like potassium, sodium, or calcium, leaks into the cell. That causes the cell to be hyperexcitable. You can imagine that can cause things like autism or ADHD or anxiety or seizures or migraine as the cells become hyperexcitable. And also, because you have to pump all that out again, you use up your energy, and so it causes a mitochondrial defect. Not because you can't make the energy, but because you use it too quickly. So how do I treat these cation leaks? Well, certainly mitochondrial targeted therapies to try to help the cell as much as possible to pump all that out so the cell is less hyperexcitable. And then there's many different ways magnesium helps, and that's really part of the mitochondrial pathway. But I'm very careful to use high-dose magnesium in these patients. Also zinc I use, B vitamins, and other things. So supplements it depends on the exact gene, but there's also medications. And I use the medication dependent on what the gene is. And so there's many different medications that work in different ways. GABA is a inhibitory neurotransmitter. So when you give GABA, you decrease the excitability. Benzodiazepines do that, and patients with autism generally do get better on those, but they have a lot of side effects, which is why I have it in italics. Glutamate is an excitatory neurotransmitter. You want to decrease that, and memantine and amantadine decrease that, and they're sometimes used in autism. Serotonin is another excitatory neurotransmitter sometimes. I'm sorry, it's an inhibitory neurotransmitter more than excitatory, and so you want to put more serotonin into the synapse. So SSRIs like Zoloft, or SNRIs like Cymbalta, or a tricyclic like amitriptyline are ones that can use here. But you can see these are pretty common medications. I also channel acting, that's Bumex, that's Diamox. A lot of the medications that people use in autism work on channels. It's just that people didn't realize that. And a lot of the supplements people use in autism, all of these have been used in autism research before, actually act on channels. But again, I mean, when I treat it, I know exactly which channel it is that's affected. Okay, most of the rest of this is going to be on mitochondria because that's really the easiest of the treatable pathways, and the vast majority of people with autism have mitochondrial problems. There are multiple papers by Dr. Fry and Rosignol that can show that. I'm not going to show the data here, though if you go to the website and say see the data, you can actually see it. This is on the neuroneeds.com website under the condition autism. These are all nutrients that there have been studies that have shown that giving this will help someone with autism. Oh, what is all that noise? It's interesting. And so this here are ones that were found in autism, and these all work on the mitochondria, and therefore they might be helpful in autism. And so, but there's no study that has shown that they are or they're not. If you go to the website, you can click on any one of these and it will actually go to the data that is, to prove that there actually is something there that you can look at the study yourself, or your doctor can look at the study yourself. And that's the landing page there. And by the way, everything that is underlined here is in our Mita cocktails. And that's in a separate product. Okay, so a lot of times people are wary of using a product that has 40 different active ingredients in it because they're afraid that, well, there would be the side effects of all 40. Actually, I find that there's far fewer side effects when you use a combination product than you use individual products. This is what you have seen before at the Krebs cycle, like in school. And this is the electron transport chain. So those are the things that you learned about in school. This is what it really looks like. This is a false color. This is an actual photo from an electron microscope of the electron transport chain and the Krebs cycle. They're really integrated together. And electrons start in one end and they go all the way through to the other. This is crystallized on the membrane. It is an assembly line and everything gets done. These things aren't just floating around and then you have to go find the next one and find the next one. The electrons just go straight through. Here's an assembly line from Ford Motor Company at the turn of the century and from Hyundai at the turn of the next century. Imagine getting this guy right here. What if you gave him his vitamins and he worked twice as fast? You're not going to make more cars. You're just going to have chaos. Same thing as if you gave more electricity to the machine in the middle here. To make more energy, you have to energize the entire pathway, not just one thing. So giving 10,000 times the recommended daily allowance of one thing, which is what a lot of people do, and see how it works, it's really more bound to fail than anything else. So the combination products work much better and they have fewer side effects. Here are some of the solutions that I've come up with. I'm a officer of NeuroNeeds, so I have a conflict of interest with it. Spectrum Needs come in 2 flavors, and it is a powder that you mix in with water or milk or juice or a smoothie or anything that you want. And it has all of these things in it here. It's a high-powered mitochondrial cocktail, plus a multivitamin, multimineral, plus a lot of these things that are in here work on the other pathways. Methylation, I didn't show you. It's a secondary pathway. Inflammation's another pathway. Amino acids, and there are many other pathways as well that are kind of minor pathways in autism. This is a very similar product, but it comes in capsules. I recommend this in any kid that can swallow. And these are the flavors to be able to take for kids that can't swallow. So around age 10 or something like that is when most kids can swallow, at least by 12. I do recommend switching to Energy Needs. It has a few more things in it, and it's dosed more for older kids or adults, and not for younger kids. So it has a little bit more of this, a little less of that. It's more targeted for an older population. It's also easier to take. You don't have to mix it. You just swallow the capsules. And it's a little cheaper because it comes in a month's supply for an adult instead of a month's supply for a child. And then here, there's the usual caveats that you need to discuss it with your physician, that they're designed to be safe, but I can't know every single patient in every single situation. And they're best combined with optimized general nutrition. What you eat and what you take as supplements, both of them are important. You can't do one without the other. So you say, which one of these? Well, these are all really the same. If you can swallow this one, if you can't swallow, choose the flavor that you want. Those are powders. The powder is either in a capsule or the powder is in the container. The powders and the oils don't really mix together. You can't really get good absorption. The oils need to be given separately to get good absorption from the intestines. CoQ is probably the single most important of all of the chemicals in my mind. It comes in 2 different forms, ubiquinone, which really is not that bioavailable, and ubiquinol, which is far better. If you go to the store, it's almost all this. But it won't say this. It'll just say coenzyme Q10, or it'll give another name. There's a lot of synonyms for this. If they put this in it, which is more expensive, it will say this all over the label. It is about twice the price, but it's 5 times more helpful. So it's actually much cheaper to get the same blood level. So I strongly recommend that you take a CoQ that's in ubiquinol. Now, not all ubiquinols are the same. There are some ones out there that are better than others in terms of bioavailability. The best ones are the QHCFs. And this one that we private label is in limonene oil, which is the oil of lemon peel instead of in soy that most of these are in. This is the size. They are things that you have to swallow. I know not everyone can do that. Another company has a liquid CoQ. It's quite expensive and the taste is only so-so, but they did a pretty good job of doing it. It's CoQ itself tastes bad. Most of my patients that can't swallow, they take these small little gel capsules and they cut the end with scissors, and then you squeeze it out and then you try to flavor it however you want. It's about half the price of the liquid if you do it that way, but it takes more work. The omega-3s are also oil. Many of you probably have children on omega-3s. Many of you are probably on omega-3s. Many doctors are recommending it now. It's good for heart health. It's also great for skin, skin, hair, and nails. It's good for kidneys, blood pressure, blood sugar. It's really, omega-3s are very important. Our cavemen and cavewomen ancestors had a lot of omega-3s they got, but in our modern diets, unless you eat a lot of seafood, you get almost no omega-3s, and if you get seafood, you're getting a lot of mercury. And so most people today, really, unless they eat a lot of seafood, are very deficient in omega-3s. So very important for brain growth. The omega-3s that you get from fish oil are good for everything except the brain. They do not cross the blood-brain barrier. The liver needs to convert it from a triglyceride to a phospholipid form. You give some fish oil, the liver will convert some of it. You give a huge amount of fish oil, the liver will convert some of it. More fish oil doesn't mean more conversion. The liver can only go so fast. So people give high amounts of fish oil. That's great for the heart. You need it for the heart. It's not getting into the brain. If you want to get into the brain, you use krill oil. Krill oil is more expensive, and the amount of krill you need for cardiac health is rather expensive. So I recommend to take fish oil and krill oil, because they're used for different things, or if you get like omega needs that we make, they're mixed together. Also in omega needs, what we do is we add phosphatidylserine from sunflower seed oil. So there's very little sunflower seed in there. It's chemically removed from there. Phosphatidylserine is another phospholipid that's needed for brain growth. Goes directly to the brain. So this is a krill and fish and sunflower seed mixture. It's really 3 products in one. That I also recommend for anybody with a neurodevelopmental disorder, including autism. Okay, the synapse is where the nerve cells meet. You know, the nerve cells don't actually touch each other, but the impulse goes down the axon, the wire, and it goes down to the synapse. It crosses over as a neurotransmitter to the next neuron and then fires. And here's the blow up of this little area. This box here is here. And you can see that neurotransmitters are released and they go to the other side. Now what this doesn't show is some neurotransmitters are excitatory, make this nerve fire, as some are inhibitory, make this nerve not fire. The inhibitory neurons are always on. It's like the kid and the cookie jar. Mommy, can I have a cookie? No. I want a cookie. No. Cookie. No. Cookie. No. Cookie. No. Cookie. Okay. That cookie is gone. Okay? The inhibitory neurons have to fire all the time or the excitatory neuron will fire. The inhibitory neurons run out of energy first. So people say, why, if he doesn't have any energy, why is he running all over the place? Why does, because the inhibitory neurons ran out of energy and the excitatory neurons are just going willy-nilly on their own. So things like autism and self-stimming and anxiety and seizures, migraine, ADHD, that sort of stuff, hyperactivity, those are often due to an imbalance between excitatory and inhibitory neurotransmitter. Now, GABA is the most important inhibitory neurotransmitter, and the GABA interneurons are particularly sensitive to mitochondrial dysfunction. So one way to help this imbalance is to increase GABA. Another way is to decrease glutamate, and we've talked about that with some of the NMDA inhibitors like memantine or amantadine. Another way is to help serotonin, which is another inhibitory neurotransmitter. And we have another product called CalmNeeds. This is a different product than the other ones. The other ones are products that you take to help the brain, help it heal, and then the brain has to heal. It takes time, 2 to 3 months to really get good effect. At 6 months, you'll notice much more advantage than you did at 3 months. Calm needs is something like aspirin. You take it because you want to feel good then. Kids going crazy, you give them calm needs, they should calm down within 20 minutes or so. It has GABA in it. It has L-theanine, which is in teas. It's in a lot of products, but it's in tea, and it makes the GABA work better. It's kind of the thing in tea that makes it soothing. It has 5-hydroxytryptophan that increases serotonin. Magnesium is a really good inhibitory. It helps GABA as well as decreases glutamate at the same time. And then B6 helps with GABA. Half of my patients that take calm needs take it in the morning. For school anxiety, test anxiety, social anxiety. They take it in the morning. They tell their mom, they tell me it's a placebo, it's not doing anything. They don't feel any different. But their mother says, yeah, but he doesn't want to, he wants to go to school now and he's not afraid of tests anymore. And when I take him off of it, he's afraid again. He don't feel any different. You can take a math test, you can drive a car on calm needs, but it does kind of mellow you a little bit. The other half, if you have trouble falling asleep because you're worried about the day and thinking about all the stuff that happened, calm needs can kind of take that worry away a little bit. It's not a hypnotic. It doesn't make you sleepy, but by reducing anxiety, some people take it before bedtime. Okay, so I show my photo here, not because I like my photo being shown, because I don't. It's because I want to make sure that these are my recommendations. And then a high-powered mitochondrial cocktail, either spectrum needs or energy needs, and there are a couple other brands out there that are good. Ubiquinol form of CoQ, which would include Q needs. I have some more information here that, details about blood levels to check, what blood levels I suggest. An essential fatty acid supplement, particularly an omega-3, krill oil for brain, fish for heart, both of them together, particularly either taking 2 different supplements or a supplement that combines them together. So these are the 3 major recommendations here. Consider taking additional zinc. For hyperactivity and other things, insomnia, anxiety, consider a GABA supplement, and those are the things in calm needs. There are other brands out there. Diet and supplements are both necessary. They're synergistic. If you have a really bad diet, you're going to have a hard time treating it no matter what you're having because your diet is not helping. On the other hand, you can get more in a supplement that you can get by eating the entire salad bar. So diet alone won't do it. They both are necessary. Genetic testing, because even though these are for the major components of autism, find out why your child has autism and if that can be treated specifically. I do 2 different things. I treat autism as a general autism plus anything like hyperactivity or insomnia or anything like that. And then as a general rule, these things work in most people. And then I figure out, and in your child has this gene, so let's try this as well. And then again, this is the get out of jail free thing. These are general recommendations. Every child is different. They're not meant to contradict your physician's advice, and discuss all treatment with your physician. Okay, running out of time now. I just wanted to, autism is a highly genetic condition. There's hard science behind that. My showing a de novo mutation in 1/2 of the cases does add something to it because other studies had far less de novos. But the fact that autism is genetic has been known for some time. There's also a very powerful environmental factor. Regressions. There's not as much evidence for the environmental factor, but as physicians that work in a lot of your families, you know that the environment makes a difference. And it's not just engraved in stone in the genetics. Also, the fact that autism is exploding in terms of how many kids have it. And our genes change only very slowly. I have a question mark on that I'll show you later. But our environment is changing extremely rapidly in multiple respects, so most people think it's probably an environmental reason as to why that autism is exploding. These are facts. These are just, we have to think about these when, when we make treatments and we figure things out. And ignoring the environmental factor, as a lot of people in the universities do, is not helping. And ignoring the genetic factor is also not helping. So, de novo mutations, are they increasing? Maybe because of cadmium or changes in our food supply like folate, or maybe it's just that they're always have been there, but in a permissive environment with all of the toxins around, it can cause disease, while in a less permissive environment, like a more older environment before we started doing all this stuff, that the same mutations were able to get by without any problems. And then there's a lot of different chemicals and everything that that are there that affect mitochondrial function in particular. There's a lot of mitochondrial function. Certainly, there are genetic components, but there's a lot of environmental components that cause mitochondrial dysfunction as well. And here is the that's how to reach me. If you're interested in having your child genetic tested, I would do that before you actually do the test, because it's really only neurability, it's really only Variantix at one lab that has the capabilities to do what I just showed you. In other ones, you're basically working on what's on the lab sheet only. That's the website and the email to reach the coordinator, Liz. And then there's other information here. And then this is a published information on genetic testing and autism that I wrote, and that's in addition to the one I showed you in preprints. And then if you're interested in the NeuroNeeds products, you can just go to the neuroneeds.com website and learn all about it there. And then these were my major collaborators in terms of the autism data that you have. So thank you very much.
Sorry, I had to unmute myself. So we'll take the next couple minutes to answer some questions. If we don't get to your question, later on, I can respond to it on Facebook. And by the way, you might want to check out, we not only have our NeuroNeeds Facebook page, but we also have a NeuroNeeds Instagram, YouTube, which this will also be on later. You might want to check back later today or tomorrow. We also just added a NeuroNeeds TikTok page. So if you like TikTok, we're going to be on there. So you might want to check that out. Okay, so for some Q&A, let's start here. Are you familiar with autism and mTOR gene?
mTOR. Yeah, mTOR is one of the, it's involved in, let me get the pathways here. What was that? It's involved in the redox state. mTOR is one of many genes in the redox state. So I can't answer specific questions about, but yeah.
Can we get a copy of these slides? As I said, this will be available on the Facebook page and also on YouTube later. So you can go through this and take your time and write down anything you need. Somebody asked the same thing. Somebody had said that their child has been vomiting for the last 6 months. And has lost 74 pounds. Cyclic vomiting, just as a
cyclic vomiting is not that uncommon in autism. It could be cyclic vomiting if you have episodes of vomiting and then you have episodes in which you have no vomiting. Mm-hmm. And so a lot of the genes overlap, particularly the cation channels in the mitochondria.
That's really all the questions that I see. As I said, if you want to watch this and later at your own time and you have questions, you can always leave the questions in the comments and we'll try to get back to you. But besides that, I think we're good. Thank you so much, Dr. Bowles, for doing this for us. We appreciate it. And yeah.
You're welcome. Okay.
Thank you.